So good evening all. Actually, mic was off. Actually, I was looking for whether you can see my screen. Fine. Oh, okay. Okay. So this is second last class. So we'll meet again, and then most probably some other circumstances, but not sure about whether we'll meet or not. Anyway, so let's start today's session. So as you are well aware about me, we have already uh, discussed several things throughout several sessions, right? So today's sessions is basically based on week 11 assignment. So before that, just to like to brief about your these things that this NPTEL program and examination pattern. So basically, you are going to get 75 question. Okay, you are going to have an exam of that will be calculated from 75, and 25 will be from your weekly assignment that you guys are doing. Now you are having 12 week, right? So all 12 week you are going to have assignments, out of which most probably 10 will be calculated, and out of that whatever you will score out of 25, apart from that the rest 75 you have to give examination. That will be basically mock MCQ or MSQ based question. Okay. Apart from this, you need to score 50 plus in order to get passed, because you are getting 25 bonus mark, and it's not difficult. We are just discussing very basic things, right? So you need to score 50 to get your certificate, and apart from this you need to score good in order to make your CV strong, and whatever for whatever purpose you are doing. Some might be doing for credit purpose, so that you can get like out of 2 credit or whatever it is. Okay. If you have any questions, you can just ask me just after once I finished all these slides, because there are not much today to discuss, because I find there are things, but it's pretty simple things, so it will not take much time. Then after that, whatever doubt you have from previous classes or whatever, we can just go one by one. Okay. Okay. So let's start with the class. So here is your first slide. Okay. So basically today we are going to discuss what we have left from the next previous classes, that in our circulatory system we have several things, right? So it is mainly regulated the systematic flow of blood circulation throughout our body in order to provide oxygen, in order to provide nutrition, and in order to provide a hydration in order to survive, right? So now if you see, the first question is that the gas exchange happens at the interface of alveoli and capillaries due to the difference in which gases, or difference in which properties. Okay, so see, in the alveolar wall it is basically lots of oxygen, because it is a part of our lungs, right? So and lungs is filled of oxygen as we inhale and exhale. Okay, so there is a rich concentration of oxygen inside the pulmonary vacuoles, or okay, and in the vein, or in the vein, or in the in the blood capillaries, basically the deoxygenated blood that is there to purified in the lungs are there in that particular, that they are flowing through the blood capillaries. And why capillaries, why not veins, why not arteries? Okay, see, the thinner the segment is, more will be the surface area. Okay, so blood capillaries are the structure that basically increases the surface area. And since the blood capillaries increases the surface area, so blood capillaries possesses a longer area of exposure to the alveolar wall, so that more amount of the membrane will be in contact with the alveolar membrane, and more will be in the contact, more amount of oxygen can be exchanged. Now why it is exchanged? Okay, these all things in our biological system, actually in the first class we have talked about one point, that is homeostasis, right? And from there till today there are lots of classes and lots of things, but everything is related to a point, that is your homeostasis. Okay, so here what it is basically maintaining is the osmolarity of gases, and always things flows from higher concentration to the lower concentration. Similar to here, the alveolar, pressure will be the right answer, because there will be a difference in pressure of oxygen. Okay, and first of all there is a osmotic gradient. Pressure will be the answer, but there will be an osmotic gradient that is created between the alveolar wall and capillaries, and after that the blood flow is occurred in such a way that there is difference in pressure of gases. Okay, CO2 it is more in the capillaries, oxygen it is more in the alveolar wall. That results in the exchange of gases. Okay, now the second question is that which of these has the higher elasticity? Now see, artery is the structure that portrayed or that balanced lots of pressure. Okay, because when our heart is pumping blood with lots of pressure, because it needs to pass to all the body part, it supplies blood to all the organelles. Okay, and if suppose you are putting water in a garden, okay, so you need to squeeze the pipe so that the blood flow will be the water flow will be in the high pressure. Okay, so similarly, okay, I can read the question is that which of these has higher elasticity? The question is asking about higher elasticity. We have options: arteries, veins, one, let it be capillaries, doesn't matter, right? So the thing is that what happened in the artery is that artery is the structure that maintains and that sustains a lot of pressure. Okay, yes, arteries definitely, but the thing is that it sustains a lot of pressure, right? So in order to sustain a lot of pressure, it needed to be thick walled. Okay, so that's why there are lots of elastic connective tissues. Okay, and similarly in vein it is thin walled because the diastolic pressure is less. However, the systolic pressure, that is squeezes to supply the blood all across the body, is much higher as compared to vein. That's why the elasticity in the artery is much higher as compared to vein, and in capillary it is very less, because it is the purpose of capillary is different. The purpose of capillary is to increase the surface area. Okay, purpose of artery is to sustain the pressure. That's why the elasticity is more. Okay, and similar, the purpose of vein, why it is much thinner, because so that more amount of deoxygenated from all across the body can travel till the left atrium in order to get till the right atrium in order to get purified. Okay, so these are the things. Now The next question is, okay, now the next question is regarding the centrifugation of blood. So if you'll find the first figure, this is just somebody taken 5 ml of blood, kept in a appendum and let it be centrifuged. Okay, so once it is centrifuged, you will find a yellow color things as shown in this figure in B. So you will find that is basically serum. Okay, and let me tell you that in biological systems, when somebody is like, what is serum basically? Serum is a protein. Serum is protein, right? And serum is basically nutritional supplier to all the cells to grow. Okay, now the blood cells and serum comprises the entire blood along with the plasma. Plasma is basically water plus serum, right? And all the blood cells are there. Okay. So the question is that if blood is centrifuged, okay, now the thing is that why it is like, it is basically plasma. Okay, so now what plasma comprises of? Plasma comprises of water, nutrients and serum, right? And serum is basically protein. And mind you that when you will find any animal cell culture and all these things, serum is one of the most costliest component. Okay, because it is the power bank that provides the nutrition, and without nutrition cell cannot grow. Okay, so now if you will centrifuge, you will find 3 layers like yellow, white and red. Red one is your RBC, white one is your WBCs, and in yellow you will find your plasma, right? Now if somebody asks what will be the layer separation of layers, okay, so you will find here in this figure that it is granulocytes that is below the layer, below all the layers. Why granulocytes? Because they have granules, they have proteins. Okay, and in the in the upper layer it is the lymphocytes and monocytes. You need to make it clear, and rather you need to have that idea that the layer of separations. RBC, it is very heavy because of the metal ions and all, hemoglobin and all. So RBC is one of the heaviest cells, so it pellet down quickly. Then you will find your WBCs, and then you will find your plasma. Okay, now let me ask you one simple question, that suppose I will collect blood samples and keep it for without centrifuge. I'll just keep it in the room temperature. Okay, so will you find this kind of structure? Anybody? What will happen to the blood if just you collected blood and just keep it in somewhere? So will you find without, will you be able to see plasma without doing centrifugation? The simple answer is that okay. Actually the thing is that you will definitely see, because see, what is centrifugation? Yeah, true. And the reason behind this is that, what is centrifugation? Centrifugation is basically application of centrifugal force in order to precipitate or in order to pellet it down, right? And now centrifugal force is basically a multiplication of gravitational force. It is basically mv square by r, right? So centrifugal force is just equivalent to your, what is the unit of centrifugal force? You will find it is in terms of G. In your classes you might have seen just centrifuge at 12,000 G, centrifuge at 13,000 G, 5,000 G. That simply means 5,000 times of the gravitational force. Okay, so the centrifugation is understood in terms of gravitational force. So if I am keeping it at my room temperature, just I am applying gravitational force, right? So it might be 5,000 times lesser, it might be 10,000 times lesser, but I am applying some amount of force through gravitation, and that will pellet it down, though in very less amounts, and it will take so much amount of time. Okay, so the yellow color part will be your plasma, right? Not platelets. Yes. Yes. Yeah. And entire WBCs is a part of supernatant only. There is no pellet. WBC is not pellet. I'll make it clear. Even RBC is not pellet. Okay. There is no pellet. Pellet, yeah, you can tell RBC, some amount of RBC or heavier RBC will get pellet down in bottom. But it's not like that it will pellet down. Okay. You will find, you can say it is a density based separation. Okay. Because some amount of liquid having lots of RBCs, if you will do it practically, you will find this structure. You can see the images, that is what is practical one, realistic one. You will find such kind of structure. Okay, it's not pelleted down. You will find the liquid portion, because see, it's not about plasma and cells. There are also waters and all these things, right? So some amount of, you can find, if you see the tube, in the bottom you will find more darker colors. Okay, however, yeah, yes, and even if you will find, as you asked, the WBC layers, I have just shown you in the top panel. You will find lymphocyte and monocyte. It just overlapped with the plasma because of their less density. Okay. So this is like this. It's not like that you can separate it. It's not like that you will not be able to separate, but you need to be careful during separation of lymphocyte, monocytes, and even granulocytes. But RBC will be in the bottom layer. Then you will find your WBCs, and then you will find your plasma. Okay. It's not like that RBC will get pellet down. You will find the layer that contains maximum amount of RBCs. It might be along with the cells, live cells, right? But it will not get pelleted just like bacteria or yeast. Yeah. Yes. Because of the concentration, because of the more cells get pellet down, right? Yes. Yeah. If the question is like that, then definitely. Yes. And somebody is saying that the question number 3, the answer will be platelets. Not platelet, it will be plasma, because plasma has no cells, right? And you will find it is separating based on the density, right? So that is the point. Platelets are more heavier, rather. They will be in the bottom, even below RBCs, most probably. Because platelets don't have nucleus, right? So you will find above RBC or within RBCs. So separating platelet is also a bit tricky task. But you can separate, because platelets basically coagulate. Okay. Mr. Madhavan, can you just turn your video off? Okay. So the next question is that which part of blood plays a major role in clot formation? Okay. Now, if you'll find clot formation, there are several factors, clotting factors, along with proteins, along with blood cells, that is your platelets. Basically responsible for, so question 4 is platelet, somebody is telling, right? Yeah, it's platelet. Platelets along with the proteins like fibrinogen. There are a series of protein, and I expect all of you to get remember rather, because it is important at BS, bachelor's or master's level. After that, you will not find any need if you are not in immunology or such field. But in this level, you might find it more useful, because question comes, and there are two pathway rather. One is direct pathway and one is indirect pathway, where basically platelets along with, there are several factors, around 13 factors, starting from fibrinogen, fibrin, fibronectin and all. Even I didn't remember, because I have also left studying these things years back, right? So you need to find it out. And the thing is that the clot formation is basically formed by platelets and protein clotting factors. Okay, so you'll find, if I will make you clear this image, that you can find that there is a gap. So there is a blood vessels broken, there is a gap in the blood vessels, and that, so that the RBCs and platelets all just oozes out. Now the purpose of the thing is that it needed to be closed, and for that, initially there are several clotting factors will come, and further they will perform the clotting. Okay, and basically platelets are the cells that perform an active participants, you can say, in this process of blood clotting. Okay. Now the next question is that, okay, now see this question. Somebody have turned your video. Can you please turn your video off? Guys, can you please turn your video off? Okay. So now question number 5 is that, if the flow in a blood vessel is F and the radius is double, then what will happen to its flow rate? Okay. Now see, I think many of you are from biology background, so it's a bit tricky for you guys to understand, but you can find. Okay, so the thing is that, see, now if you let me discuss about what, let me discuss about the equations. Okay, so here you will find the resistance to flow. Like suppose there is a, I'm just generalizing the concept. Suppose there is a pipe. Okay, so if, or you can, let's take it blood vessels. Okay, so if you will increase the blood vessels, the volume that will flow through will decrease, right? Because there is a large area. And if you will decreases the volume, or you will decreases the radius, then the pressure will increase. If the same pressure is there, then it will, the volume will be, or the flow rate will be more. Just, it's very general, you can just realize. Okay, but you will find this equation, then the volume or flow rates is, there is a formula you can find, pi and then pressure difference and then radius square divided by 8 into viscosity into length. Okay, so basically I can just directly tell, as basic as it is, that the flow rate is 4 times proportional to the radius. So if radius will be 2r, then it will be 16r, right? So it will be 16 times more. But if you try to understand the concept, then even I have to make you clear about the equation, that is, what is eta. Eta is the coefficient of viscosity or something. So, right. So there are several points now you need to understand that what we are doing. Suppose we are increasing the radius. Let me clear. Okay. Suppose we are increasing the radius. So radius will be 2r. Okay. Now if radius will be 2r, okay. So the cross section at a particular point is equal to pi r square, right? if you can see my crosshair, this particular area is your cross section, right? So this area of this cross section will be pi r square. Okay, so this will be 2 times. Okay, now you need to find the volume rate. Okay, so the volume rate is proportional to the cross section as well as the length, the entire cylindrical volume. Okay, so the cylindrical volume is basically directly proportional to the 4th power of the radius. Okay, even with the equation we can just define this, but it is pretty easy, or it is pretty general. Just you remember that in blood system, in vascular system, if we double the radius, okay, basically the radius is directly related with the 4th power. The flow rate is related with the 4th power of the radius. So if we increase the radius double, the volume will increase 4th time, that is 2r. It is the 4th power of the 2r, that is 16th r. Okay, similarly, if you will increase the radius 3 times, then again the radius will be 3r, and it is directly, if you will calculate the volume rate, or it will calculate the flow rate, it will be 4th time of that particular radius. Okay, so in vascular system, the flow rate is basically proportional to the 4th power of the radius. Okay, so just make it clear. Okay, and if you are going to discuss this in details, even I need to make it clear to you guys, and it will take a lot of time because we need to understand the individual equations, and I don't think you guys need that. Okay, so just make it clear that the radius is basically, it is the 4th power of radius is related with the flow rate. Okay, now there are 2 questions. Okay, what is the standard systolic and diastolic pressure? I have discussed in the last class, so we can just quickly skip this question. So the systole is the pressure created by the heart in order to supply the blood throughout the body, and diastole is the pressure created by the heart itself in order to make the blood pass into the lower heart chamber. Okay, now we all know that the normal blood pressure range is 120 by 80. You all know, you all should be aware of, right? So the normal range is basically 120 by 80. So the diastolic pressure is 80, and systolic pressure, the pressure required to the pressure created by the heart in order to supply the blood throughout the body, that is definitely more, because we are putting pressure in order to supply blood. And diastolic pressure is just to supply blood from the upper chamber to lower chamber, and that is also easy, right? Answer will be 16 here. Okay, it is 4th time or 4th power of the radius square. Okay, so if radius will be 2r, the flow rate will be 16r, that is 4th power of 2r. Okay, now the 6th and 7th question is that the systolic pressure is basically the pressure created in order to supply throughout the body, so the pressure will be more. Okay, and that is basically considered to be 120, which is normal. Okay, so 120, and in your option, anytime when you are attending MCQ question, you need to be careful, right? Because see, there in the option you have 120 to 140, and I guaranteely say 140 is not a normal blood pressure. Okay, 140 is high blood pressure. You can find in your parents or elders, you will find this kind of blood pressure, and that is not normal. Okay, so 140 is pretty high. You need medications, rather I can say. Okay, so that is called hypertension. But still, if you find your options, there is no options. You have to go for 120 to 140 mmHg. Okay, and similarly, the diastolic pressure, it will be in between 60 to 80. Okay, because 80 is your normal. So mind it, the systolic is 120 and diastolic is your 80 mm or mmHg. Okay, and I think you all are aware of these things because this is pretty basics, right? Now question 8 is the carbon monoxide poisoning happens because of the interaction with what? Okay, now the answer will be hemoglobin, and I don't care about the answers, rather I need you to understand that how it is happening, right? So you all know that hemoglobin is an oxygen carrier, and how it carries oxygen? We have irons. Okay, it is basically a porphyrin. There is a structure, you can find the structure of hemoglobin from book, and you will find that there is a positions in the upper panel and the bottom panel in both areas where oxygen can bind. In one hemoglobin you will find there are 4 oxygen can bind. Okay, now what happened? What is the role of hemoglobin? It binds to 2 molecules. It's not only oxygen, right? It binds to oxygen, it binds to carbon dioxide. Mind it, carbon dioxide. Okay, now what it does, as I told in the first slide, in the capillaries the blood transferred, right, exchanges with alveoli, right? Now that is occurred. It's not like that oxygen directly transferred, it is through hemoglobin, because hemoglobin is the oxygen carrier, right? Hemoglobin will release carbon dioxide and it will get binded with the oxygen, and both are beneficial for us. It's not like if carbon dioxide binds to hemoglobin it is not beneficial, it is also beneficial. But the point is that when carbon monoxide binds, okay, when carbon monoxide is getting binded with the hemoglobin, it will form carboxy hemoglobin, right? Somebody have made messes. So it will form carboxy hemoglobin. Now the thing is that neither carbon dioxide is getting binded nor oxygen, and if there is no carbon, that means it neither purifying our blood nor it is decontaminating. Both is not happening. Neither it is purifying, neither it is depurifying, right? Neither it is taking away carbon dioxide, neither it is carrying oxygen. Rather it is a third party or third molecule, it is a foreign molecule, that is basically carbon monoxide, that is getting binded. And that is the point, is that it is results in the carboxyl hemoglobin, and that is not carrying neither oxygen nor carbon dioxide, and that will result in anoxemia. Anoxemia means lack of oxygen, and it will lead to suffocation, it will lead to severe allergic reactions, because there is no oxygen, you will find suffocations. Okay, so the thing is that carboxy hemoglobin, carbon monoxide is basically replaced the oxygen and carbon dioxide. That is what we don't want, because we want it to getting binded with either carbon dioxide or oxygen, so that it can get exchanged at any point of time. Okay. The last question is there. So blood vessels are mostly made up of it is, I think we have already discussed several times, smooth muscles will be the answer, because smooth muscles are a part of several all the vital systems. Okay, so this is mainly a part of blood vessels as well, and the blood vessels basically comprises different type of cells. One is your smooth muscles. Apart from this, you will find many connective tissues as well. Okay, so that it cannot be rigid, it need to be flexible. Okay, so that is the point. Smooth muscles are very much essential part of all the vital system along with the blood vessels as well. Okay, and the unit of hemoglobin quantification in blood test report is gram per deciliter. Okay, if you have ever done it is gram per either liter or gram per deciliter, because gram is one is gram is what it is called, it is considered one of the standard units, right? So gram per deciliter is basically found in blood report, and if you Consider the standard value, it is around 14 to 18 in male and it is 12 to 16 in female. Okay, now the question is that why it is higher in male? Anybody? It's not not arguable, but the thing is that it is there, it's 14 to 18, we can't do anything. But yes, but if we'll, yes, yeah, that I agree. But why that adaptation is occurred in that way? Why RBC is more in male? Because see, as we discussed, everything can be related to homeostasis, everything can be related to evolution, everything is evolved in a way that have some beneficial role, right? So it must evolve, and it occurs, it is because of, it's not like that because of the thing it is evolved, it is like it is the condition that permit the males to work more physically. This is the point. It's not like that we can't say like that males are more physically active, that's why they have more hemoglobin concentration. Rather, they have more hemoglobin concentration, they have more RBC count, that's why they are able to more physically active. That we can conclude. Okay, but evolutionarily, if you find that it is evolved in such a way because it has some reasons. Okay, and we need to find out the reasons. You can find it out. Okay, so it is pretty straightforward questions today. So I think all of you are clear with the question. Apart from that one, that is the calculation was there. So that I think we need to go to some law, right? That is basically flow of liquid. It is there is a chapter in 12. So you can just even I can find it out. We can discuss if I'll find it out next class, or you can just remember that it is related in that way. Sure. Just yeah. Okay. See, if the flow, can you mute? Okay. Okay. So the question number 5 is that if the flow in the blood vessel is F and the radius is doubled and the pressure is kept same, then the flow would become what? Okay. Now the thing is that we are see, if you will directly consider the formula for the volume flow rate, then it is not tough for me to make you explain, right? So basically the formula for volume flow rate is F, that is equal to rho 1 minus rho 2. Okay, and that is equal to it is defined as pi, then the difference in pressure, that is rho 1 minus rho 2 r square, okay, divided by 8 viscosity constant, that is eta, and length, that is L. So the formula is and yeah, I can just explain you guys, it's not that tough. These equations are not that tough. Now it come to me. See, the R is basically resistance of flow. Okay, so the formula for R is that 8 eta L divided by pi R4. Okay, so it is defined. You can find the derivation. I don't know, speaking frankly, because I have not gotten that depth. But what I can interpret from this is that eta is directly proportional to viscosity, because eta is the constant of viscosity. Okay, now the resistance का formula है वो है 8 eta L by pi R4. See, resistance का जो formula है, resistance is opposing force, right? So resistance का जो formula है, that is 8 eta L by pi R4. Okay, now हमको what we want to find out, we want to find the velocity, we want to find the volume flow rate. Okay, so that is against the uh, resistance, right? So volume flow rate is 1 by R. Okay. Now 1 by R means, if you will just consider that particular aspect even, if you will not go into the details, just opposite of R, you can just make it 1 by 8 etal by pi R4. That simply means pi R4 by 8 etal, right? It will be like this, right? Just make it reverse. Okay. Yeah. Even and the entire formula is like pi pressure difference. Don't go into that. Just first consider that it is pi R4 by 8 etal. Okay, right? And the pressure difference is there, right? And we told in the question it is mentioned that the pressure is constant. Okay, so that factor is not going to affect. Now, what other factor can affect? We have viscosity, that is also constant in our questions. Okay, length, that is also constant because blood vessels length is not going to change. Okay, pi is a constant. So the only thing that is going to change is your radius. And now the radius is 2r. That is in the question it is asked the radius is doubled. Right, so just put 2r in place of radius and it is the fourth power, right? So it will be 16 times basically, because apart from that all are constant, right? So so this will be in that way. Pulse pressure, okay. Pulse pressure is, how can I define that? We can define in general term that it is basically systolic pressure. Okay, see, now what we can see, okay, what is the pulse rate? Okay, so the pulse rate is basically 72 times per minute. This is basically beat. Okay, now this heartbeat is basically responsible for maintaining the cardiac cycle. Okay, now just let me tell you generally that the heart rate is basically related with your pulse rate. Okay, so if it is set 72, which is average beats per minute, then you will find one cardiac cycle will take 0.83 seconds. Okay, and within the time the entire cardiac cycle is taking place, right? Now if you will find there are two kind of pressure, one is systolic and one is diastolic, right? So the systolic pressure is the pressure that is the blood is the heart is pumping to to eject out the blood to entire body. Okay, and it is the diastolic pressure that heart is pumping in order to get the blood into the lower chamber. Now the pulse pressure is basically defined by the combination of both. You will find the both systolic and diastolic pressure will result in the pulse pressure. Okay, it is just kind of resultant, but generally the pulse pressure is mainly affected by your systolic pressure. It's not by diastolic, it is systolic pressure, because diastolic pressure is the pressure that is created by the heart itself to pump the blood into the lower chamber. Okay, but it is the systolic pressure that heart squeezes out the blood to entire body system, and that is what we can feel here, the pressure that is generated by the heart in order to supply the blood throughout our system. So that will be the pulse pressure. Okay. Yeah, you can find it out. If you find something different, then you can come, because see, you are even I am not. I am just making it think it like what it could be. Okay, I might be wrong in some days. So just if you find something different, just come to me, that we can discuss, right? Yes, yes. Hmm. Mhm. Yeah. Yeah. Mhm. So can I tell like in females the level of progesterone hormone is more, that's why it is decreasing the amount of RBC in female? Hmm. Yeah, yeah, I understood. Definitely, yeah, it can be a reason, but definitely what I want to tell, see, uh, practical, yeah, practically what can be the reason, and if you'll find, let's think another aspect, okay? So initially, just I'm making you some general idea that initially there is no haploid cells in our body. Okay, initially all cells are somatic cells only, right? Now some somatic cells modified in a way that they developed into the germ cells, right? Okay, and again within that germ cells there are some modifications occurs that define it whether it will be a male germ cells or female germ cells. Okay, and again further it will go downstream in order to more confine whether it will be a male or it will be a female, right? So what I can say, it can be a reason, there are several reasons, because see, if you put a male and a female structure, so there are several differences, okay, in starting from the embryogenic development till the biological functions, and there are so many changes, and as I always told that everything has a correlations. Okay, so it is not it will not be that appropriate. Rather, see, we can say, but we cannot justify, right? The level of androgens can be related to RBC, because we need to find out some proof in order to say that this can be a reason. And you will, I can guarantee you will find papers, right? Research papers on this. You will find definitely one or two. It's not a big deal as well, because people are working, and you will find definitely. Suppose I'll let me take you in a science journey. Suppose I want to understand the role of anything, any proteins, any markers, anything in cancers, I can find it out. Okay, because why? Because why? See, let not be cancer. Let take common cold. Okay, if it is a common cold to you, we basically related with it is because of virus, right? Yeah, but that results in entire metabolic changes in your system, right? So if somebody study from some other aspect, like if somebody will tell that, you will find, you can read, or you can yeah, practically you can study from different aspects. Somebody will read the, study from your legs and will tell it is shivering, that's why it is cold. So it is a different vision. The way you will see the research, it is like this. Okay, it is not at all like you are going to find it out. Rather, I can say, whenever you will guys come to research or like that, you will find it's nothing like, it's not like what you are thinking and what it is happening. It's everything what you want to sow, you can sow. Now the beauty in research, that how beautifully you can prove your thoughts, that I want this and this is what is best occurring in this disease. In order to sow, I can sow anything. Hmm. Hmm. Yeah. Hmm. Yes, right. Yes. Hmm. Yeah, yeah, it's true. That's what I was actually supposed to talk. Hmm. 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 Yeah, that's true. Yeah, that is the cells. I think it converted to some cells that really results in the androgen secretions, rather, I can say. Yeah. Hmm. Hmm. Yeah, that's true. That is what. And but see, that's true. That is why it converted to it somebody is male or female. But we are talking about an aspect that on RBCs, right? Now, yeah. So we can for the time being see. I would like to suggest like ki, many time we think that why it is happening, right? But sometime we need to think like this ki this is happening, that's why this result is occurring. Okay, every time we can't say ki, why it is happening, why you are here. Okay, sometime we can say like we are here so that we can do something, right? So no, that's definitely fine. You all are will be in the research in few years, so you need to ask good questions, and asking good question will solve almost half of your problems, because addressing questions